Hello, everybody. Welcome to the Ethel Browne Harvey Postdoctoral Seminar Series. My name is Lydia Djenoune, and I'm a postdoc at Mass General Hospital, and I will be moderating today with Granton Jindal, a postdoc at the University of California, San Diego. We are excited to highlight the work of our outstanding postdoctoral members. And today, Emilio Mendez Olivos from the University of Calgary and Cote from Stanford University will share their research. So each speaker will give a 20-minute talk followed by 10 minutes of Q&A. So please enter your questions in the Zoom Q&A box as they pop up. So it's my pleasure to introduce you to Dr. Emilio Mendez Olivos, who is going to be our first speaker today. So Emilio, after graduating from the Pontifical Catholic University of Chile, where he investigated spinal cord regeneration in xenopus under the supervision of Professor Juan Laren, he then joined the group of Professor Miguel Concha at the University of Chile, where he studied the evolution of asymmetry in the epithalamus of ray finfish. Now working in the group of Professor Peng Wang at the University of Calgary, Emilio is currently funded by not only one, but two grants, the ACHRI and the Cummings School of Medicine postdoctoral fellowships to explore how a diverse population of fibroblasts arises from a defined population of sclerotum cells in the trunk of zebrafish. And Emilio is going to tell us all about it now. So please join me in welcoming Emilio.
Thank you very much, Lydia, for that super nice introduction. Thank you, everybody. And thank you for attending today. I would like to start this talk by what is my interesting in science. And one of the most beautiful processes that I discovered when I just started studying developmental biology was cell migration. As can be seen in these movies, migratory cells happens to occur during development in many different contexts and in many different parts of the embryo. Here I'm just showcasing two of them, and I hope that you can appreciate how intricate this mechanism needs to be and also executed. In the lab, Wang and the group recently, when I just joined, they were interested in the development of the sclerotum. Let me tell you a little bit what is it. The sclerotum is a transient structure that is composed of the ventral part of the somite that you can see here in this diagram, and it will later subspecify and will contribute to the development of what is known as the axial skeleton, mainly composed by the vertebral body, the axial processes, but also the tenocytes that are at the edge of the intercostal muscles. So out of this seemingly a small population, we have quite a variety of different fibroblast-like cells that it will need to move, because just take a look at here. You have the notochord, here is the spinal cord. A lot of this region will have to form this entire structure. So there is a migratory component that it's not well known. Fibroblasts, probably you have heard of them because it's one of the most common cell type in our body. And their primary function has been described as extracellular matrix remodeling, either by producing or degrading in certain scenarios. Here, for example, I also like to showcase how these fibroblasts will need to eventually self-renew and differentiate. They can also help or support any kind of migratory process of themselves, but also other cell types, and also secrete a bunch of different signaling molecules to provide some context in the tissue that they are forming part of. But how do these cells land in pretty much every single organ of our body is not pretty well understood. And the migration of these cells need to be addressed. So the model that we use in the lab, it's zebrafish sclerotum. Here you can see that it's sustained by the expression of NK3.1. And in zebrafish particularly, the sclerotum that I mentioned before is composed of that dorsal and a ventral domain. And in this transversal section, you can see that at 24 hours, there are NK3.1 positive cells going around the notochord. This is a hallmark that these are notochord associated cells or sclerotum derived. They are starting to leave their sclerotum fate and acquire a commitment toward notochord enchyting cells. And this is the model that we are looking today. Let me show you this movie in which you will be able to see how do these cells migrate from 24 plus to 13 hours after. And I hope you can appreciate how through the use of this reporter, NQ2.1 driving GAL4 and the expression of NTRM cherry, how from a very defined dorsal and ventral domain, we'll have a tremendous amount of different cells populating the trunk of the zebrafish. I will play it again just because it's beautiful and I really like to look at it. And you will see the cells going toward the notochord. Some of them will align in the myotendinous junction. The fin compartment, these are fin mesenchymal cells. And this is some of the components that I label in my next image. Here, they are labeled in cyan. And for contrast, we have the expression of KDR expressing M cherry. And we have these perivascular fibroblasts, we have fin mesenchymal cells, notochord-associated cells, tenocytes along the myotendinous junction, as I mentioned before, and interstitial fibroblasts, among many others. So the skeletal zebrafish is actually the source of many different cell types. And as there has to be a specificity in the migration of these cells to give into account how do we develop properly all these different cell types. So the question that I decided to study in the lab, it's actually how these cells leave their dorsal and ventral domain, and they eventually land in their differentiation compartments. One of the first steps that we took was to study a very peculiar and pan-fibroblast marker. You will be seeing the word PDGFR alpha throughout all the literature, especially single-cell RNA-seq as a pan-fibroblast marker. All the fibroblasts will express it. But what is actually the function of this protein? Well, interestingly enough, when I did the in situ hybridization, I found a perfect match for the sclerodome compartment in between our marker NK3.1 and the expression of the mRNA of PDGFR alpha. And this can be seen better in the orthogonal view, while the match is almost perfect for the sclerotome. So there has to be a function for this gene. PDGFR alpha has been described as an important signaling pathway in other migratory cell types, as one of them, neural crest cells. This is zebrafish, neural crest cells labeled in green, and I hope that you can see how in normal condition, the cells go around the eye, but in the mutant for the receptor, the cells fail to go around the eye and develop anterior structures. Similar phenotype can be seen in the formation of the heart or the heart field that it need to migrate ventrally from the lateral plate mesoderm all the way down in zebrafish 2, revealed by the expression of HAN2. Wild-type embryo, they form this horseshoe shape at the midline, but in the PDGFR alpha knockout animals, they never fuse and they fail. And eventually, this leads to the death of this animal by day 5. PDGFR alpha signaling pathway is composed by 2 ligands, PDGF-A and C, that can bind the receptor. And upon the binding to the receptor, we'll have the activation of the tyrosine kinase domain. I have this diagram to show how the equivalence or the correspondence of the gene in the zebrafish genome with the human one. Just to highlight that we have 2 paralogs that are ortholog to PDGF-A in human, PDGF-AA, PDGF-AB due to the genome duplication that actinopterygia went through. But nonetheless, upon the activation of the pathway, we will have that many different processes will be triggered. Some cells will proliferate, some will differentiate, some will survive or will promote their survival. On the important one that we will be talking today, it's migration. So to study this pathway, we asked Dr. Xiao Yang, which they published with Susan Alras, the work in the heart field, for the line. They used this PDGFR alpha gene trap line in which the gene is disrupted by the insertion of this segment just before the transmembrane domain will lead a non-functional protein that, as you can be seeing here, it's a fusion of the extracellular domain with MRFP, completely different from the functional one. And when we stain against our marker NK3.1, I hope you can appreciate how we lost the migratory cells out of the ventral domain, which is pretty striking. This correlates perfectly well with a notochord-associated marker, which is PAX1A. Only the cell that reaches the notochord will express this gene. And you will see how it's a dosage-dependent decrease of this one. We wondered and to check if this was specifically due to the insertion or the loss of function of the gene, we decide to rely on these LOXP sites. So we inject mRNA at the single cell stage, and in the control animals receive no mRNA. We have the expression of MRFP driven by PDFR alpha, so it's a reporter. But NK3.1, you can see there is no migration, as I showed you before. But when we inject mRNA, it's hardly to see because there is no MRFP expression. But interestingly enough, we rescue the migration of the NK3.1 cells, suggesting that the lack of migration was due to the interruption of the gene. Here, you will see how do these cells move through time. Similar movie as I showed you before. This is the at the left, the wild-type animal with all the cells going to the notochord compartment, and the rest of the cell type that will arise from the sclerotom. In the homozygous mutant for PDFR alpha, you will see there is hardly any migration. And this pretty striking phenotype also correlate with the lack of differentiated cells or sclerotom-derived cells. Here, one of the population labeled by PRLP. These are tenocytes along the myotendinous junction. You can see the reduction using our reporter line. I quantify those cells and also correlate with the reduction in the staining, a decrease in the number of tenocytes. This is also true for a different population, which is fin mesenchymal cells, also derived from the sclerotom, labeled by fibrillin expression. The wild-type animal, beautiful staining around the tail in the fin fold compartment that is reduced in the heads and almost absent in the homozygous mutant. When I count the fin mesenchymal cells, I observe the same decrease as I saw for the tenocytes. Then we wonder, is this phenomenon cell autonomously, meaning it's a direct or an indirect effect of the lack of the expression of the receptor at the cell surface level? To do this, I generate this dominant negative form in which I replace the cytosolic domain with EGFP, but it will be membrane bound at different, in contrast with the gene trap line. And first, when I inject in our sclerotom reported line membrane target EGFP, I found that the EGFP cells can be found throughout all the different compartments derived from the sclerotom. But when I inject a UIS dominant negative PDFR alpha, the EGFP cells resides mostly at the ventral and the dorsal domain, and it's hardly to find cells in the notochord or in the fin fold, which I also quantify, suggesting that this is a cell autonomous effect of the receptor. So as a first summary, we have now the NK3.1 cells need to express PDFR alpha in order to reach their final destination. If they do not express PDFR alpha, they fail to migrate. One of the questions that sparked here was, what might be the ligand activating the receptor? So we screen for the ligand that I mentioned in my introduction, PDFAA, AB, and C, and due to the expression pattern, we decide to focus our interest in PDFAB. And in this transversal section, I hope you can appreciate the beautiful complementary pattern in between PDFAB expression and PDFR alpha expression, giving like a good hint that this might be the ligand activating the migration of these cells. Nonetheless, the expression of the PDFAB genes also resemble another very peculiar expression pattern, which are the sonic hedgehog target gene around the notochord, especially, but in the myotome. So one of the genes that I decided to use was patch2, and I hope you can see that there is a pretty good level of co-localization or co-expression, in this case, of PDFAB transcript with patch2 transcript. Due to this, I decided to inhibit hedgehog signaling through the use of cyclopamine to test if I might affect the expression of PDFAB. And here you can see the expression in the DMSO-treated animals. The migration is perfectly fine, revealed by NK3.1, but the expression of AB is compromised upon the use of cyclopamine with the embryo, which correlate with the lack of migration of these cells, hinting or suggesting that hedgehog might be upstream the expression of AB, and in that way might regulate the migration of these cells. We are not sure if this is exclusively the only mechanisms, but it's a pretty good lead. Then, given the fact that PDFAB, now the ligand, is affecting the migration, we reach circ and ask for a mutant animal for PDFAB that hasn't been characterized. And in that animal, as you can see by, again, NK3.1 staying 24 hours, we observe the same reduction that we observed with the lack of PDFR alpha in the migration of the scleroton cells. This also correlates with the reduction in the expression of PAX1A that is perfectly fine in wild-type animals, but is reduced and almost gone in homozygous animals. How do they migrate? Here you have the movie. Again, at the left, wild-type animal, beautiful migration, different compartments. Homozygous mutant animal, lack of migration toward the notochord compartment. But I will play this one just one more time. Now, don't look at the most striking feature. Look at the film mesenchymal compartment, which is another compartment that is interesting. The migration here is fine, and the migration here is also fine, suggesting that it might be a specificity on where the cells are being attracted by the expression of PDFAB, which makes sense because we saw the expression of AB mostly around the notochord, not at the fin compartment. When I quantify the number of film mesenchymal cells, we observe no significant difference. But when I quantify the number of tenocytes, I observe a similar reduction in the number as I saw for the lack of the receptor. So now, to summarize the ligand, we have that in our previous model with PDFR alpha and NK1 expression, we can add on top of that one the expression of the ligand in the submitting compartment around the notochord region. And this one might be attracting the cells toward that compartment exclusively or specifically. Upon the loss or in the mutant animal for PDFAB, we have that the migration toward the notochord compartment is compromised, but to the finfold seems to not be affected. Now we ask, is this signaling chemoattractant, or meaning that it's instructive or is permissive for the migration, a concept that we extract from other migratory cell type as neural crest cells, for name one. To do this, we perform an overexpression of the ligand through the use of a heat shock promoter. In the wild-type sibling, heat shock at 18 hours, we observe that this is the level of expression of AB at 24. The migrating cells are fine. When we overexpress PDFAB, of course, the in situ doesn't look that great because now the mRNA is everywhere. But pay attention how we also lost the migration from the ventral domain. This is pretty counterintuitive but at the first thought, but when you think about it, now the signal, it's being expressed everywhere. So what is used to be a gradient or a signal, a beacon attracting the cells to one specific source, now is present everywhere. So the most likely explanation might be that the cells now do not have a proper path to follow, and they get lost or entangled in this misexpression. Another approach that we decided to use to address this very same question was transplantation experiment. What we did was we took wild-type donors injected with mRNA to label them and be able to track them into a host that is PDFAB mutant, homozygous mutant. We did the transplantation at blastula stage, 4 hours post-fertilization, and the idea was to look at the animals at 24. And I'm going to show you images of just a couple of somites, the ones that are migrating at this particular stage. Here, it's a comparative. This is how the wild-type donors look like. This is the expression of CADA. The NK1 migration is fine. In the host that hasn't received any CADA cells, this is just an specific staining of the antibody inside the notochord. We observe that the NK1 migration, it's absent or it's reduced, heavily reduced. What happened upon transplantation? Look at this. We have the expression of CADA positive cells throughout these mosaic animals, and the migration, it's rescue. But interestingly enough, look at these three. For example, if we look at this one, we have the migrating cell in the orthogonal plane going around the notochord, and there is one CADA cell there. If we look at this one now, that is, it hasn't reached the same level, but the posterior one, it did, we can see that the cell, it's kind of in a more exterior part. Finally, if we look at that one, we observe the same as the first one. Here we have the migrating cells, and we have also a cell pretty close to the notochord. And also there is always a contribution to the spinal cord. What is in this other scenario in which we have a massive contribution, but there is no rescue? What is happening with this animal? We observe that the cells do not localize close to the notochord mainly. They are mostly at the superficial level here, or at the dermomyoton compartment close to the surface, or even at the dorsal part of the neural tube. When we quantify this, we found that 50 out of 54 rescue somites display contribution in this compartment, and only 4 out of 54 display contribution to the neural tube, suggesting that is when the wild-type cells contribute to this region, which is the PDFA-B region, this is the region that account for the most number of rescue somites. So
Emilio, you have 1 minute left. Sorry.
Thank you, Lydia. I'm just going to finish with my model, which is the one that I already briefly showcased before. We have the expression of PDFR alpha and KIT3.1 and AB. Upon the loss of PDFR alpha, there is no migration, but we retain the expression of the ligand. When we don't have the expression of the ligand in PDFA-B mutant, we have the expression of the receptor, but the cells cannot migrate toward the notochord. And when we perform the transplantation experiment, these are the small dots. Yellow here are attracting the cells. When do you not have cells at the proper place, you do not rescue the migration. So that being said, I would like to acknowledge my lab, my supervisor, Pang, for his great mentoring skill and also support to my research, all my lab, all our collaborators, local collaborator and external collaborator, all the funding sources, CERC, and all of you for your attention. Thank you, guys. Happy to take any question.
All right, guys. Thank you, Emilio. This very elegant talk. Really nice and elegant experiments using the power of the zebrafish. So something that makes me happy. All right, so we have a question. The first one is, when you knock out PDGF receptor A, cells are migrating along the dorsal ventral axis, but not towards the notochord. Do you expect any mechanotransduction signaling involved that induces polarity of the fibroblast in addition to chemoattractant?
It's something that we haven't explored, but it might be a possibility. I don't know if you remember the movies, but in the movies, when we have the migration toward the notochord compartment, the cells do not seem to be pretty close to each other. But I think that it's a caveat of our model because we are labeled way too many. But it's something definitely that I want to explore if there is some mechanical polarity involved, for sure.
Thank you. So the second question is, have you assessed the potential of sclerotome-derived tenocytes in an injury context?
Oh, that's a great question, and I haven't. But actually, now Dr. Arshin Rajan, she was focused on her work in injury and sclerotome-derived cells later on. And she showed that the importance of Sony-Hecho pathway for that one. But now that's work of Haley, which is working with the regeneration model. It's definitely a super interesting question, but it's out of the scope of my project.
Thank you. So there is another question from Granton, who says that it's a great talk. So he's wondering what signaling pathways are downstream of PDGF receptor A and what migratory genes are induced downstream of the signaling pathway.
That's a great question. I've been curious about that one since I started. I think that sometimes you want to go faster. But the only thing that I can comment, it's been depicted that the importance of PI3 kinase, but which genes are actually being activated, it's something that I think we should test that out in our model, especially the mutant for PDFR alpha. RNA-seq might be a great tool to use with those animals to try to tease apart which are the genes that probably are involved. I will expect to see something similar as other migratory cells, like neural crest cells or Harfield cells that need to do this epithelial to mesenchymal transition. So probably that's something that I will bet on.
So please add your question in the QA. Okay, so there is another one. somebody's asking, have you looked into the effects of PDGF receptor B?
I wasn't expecting that one, but yes, I have. And the only thing that I can show right now, I hope that you can see it. Thank you for the question. I will send the transfer later. PDGFR alpha and PDGFR B both are expressed in the sclerodome level, but when I did the experiment with the dominant negative ones, only dominant negative for PDGFR alpha displayed the phenotype. When I overexpress PDGFR B, the cells are perfectly fine, and as you can see, they can be found everywhere throughout. So it seems that it's not as important as alpha, and for I don't know what else can I say.
Okay, so I have a question myself. Did you look at the neurons within the spinal cord? Like, what happens to them when you mess up with the migration of
That's something that we want to explore, actually, because we have Artie Gekrivat in the lab. She's studying the spinal cord development properly. And that's something that it got right, because when, I don't know if I show that one, but PDGFAB is expressed in a certain population in the spinal cord. The receptor is being described being important for the oligodendrocyte development. So definitely there might be something wrong. I wouldn't be surprised, but we haven't explored it yet.
There's another question. Also, can you be sure there are any differences in the homoligand PDGFAA and heteroligand PDGFA, PDGFC? The person assumes that they would have different phenotypes.
Great, awesome question. No, but the short answer is no. The only thing that we have to address that is that the expression pattern, as you remember, PDGF-AB is expressed in the middle of the somite, and PDGF-C seems to be kind of delayed somehow. It's not exactly the same. There is this region that is shared, but it seems to be kind of in a different part of the somite. What we thought it's PDGF-AB, it's attracting the cells toward the notochord compartment, and C might be involved in the fin-fold migration, but we haven't done anything related to C yet, but it's something that I truly want to explore. Thank you for the question.
Another one. Have you looked into the effects of PDGF beta ligand, since it is a well-known migrational factor? Some people are like, the person is interested in the interaction between PDGF receptor A and PDGFB.
Oh,
and yeah. Okay.
Yeah. I think that if I get it correctly, PDGF-B and the receptor beta are well known for their role in migration. So no, when I screen for I also did the screen for the expression of PDGF-B ligands, but they do not seem to be involved in this particular process. They are known for being involved in the migration toward the hypocore and the angioblast to develop the vasculature of the fish. So, but I can't rule out their implication more than the experiment that I showed, that PDGFR beta doesn't seem to be involved. But if it's actually B acting through alpha, it's not that has been described extensively. The receptor might heterodimerize, but I don't know if I have the tools to fully address that part.
Nice. Any other question, guys? Or Emilio, is there anything else you want to add?
No, just say thank you very much to all of you guys, to the organizer for giving me this amazing opportunity. And thank you for all your questions. They've been great.
All right, thank you, Emilio. So I guess Granton can take over now. Thank you.
Hello, everyone. Thanks, Lydia, for co-moderating, and thanks, Emilio, for your excellent talk. So I have the privilege to introduce our second speaker today, Dr. Lauren Cote. Lauren received her undergraduate degrees in biological sciences and mathematics from the University of Chicago. Lauren received her PhD in the lab of Professor Peter Radin at MIT. Her thesis involved understanding the instructive roles of muscle cells in planarian regeneration. Lauren is currently a a postdoctoral fellow in the lab of Professor Jessica Feldman at Stanford University. She is investigating how cellular apical basal polarity establishment drives tissue organization along the C. elegans digestive tract as a Damon Runyon Fellow. Lauren has also received many other competitive fellowships and awards. The subject of her talk today is making and breaking one continuous digestive tract. We are all looking forward to your talk. Take it away.
Thank you so much, Granton, for that really lovely introduction. And thank you to the SDB selection committee for giving me this opportunity to share my work with you today. So I'm Lauren. I'm a postdoctoral fellow in Jessica Feldman's lab. And today I'm really excited to share with you some work that I've been doing on making and breaking one continuous digestive tract. And so I'd like to start with the observation that life really requires lumens. This is evident from the very beginnings of life, whether we want to move fluid or filter fluid in certain specific organs, or whether we want to put many of these organs together into a full organ system that really support life. And when luminogenesis goes awry, we have many different disease contexts in which this can happen, including the formation of congenital atresias and fistulas, or say in polycystic kidney disease. And so when we think about luminogenesis, all of these lumens are formed from epithelial-like cells. And so when we look at a very simplified version of an epithelial cell has an apical surface facing a lumen or the outside world. They're connected to one another via cell-cell junctions and have also a basolateral surface that faces other cells. And when we think a lot about epithelial cells or see them depicted in reviews, these cells are often depicted kind of in a vacuum in the middle of space. But we know that some of the really crucial components that go into making these epithelial cells include the apical par complex, shown here in green, and E-cadherin, which is a homotypic cell-cell adhesion molecule often found at cell-cell junctions. And so these are going to be the two molecular players I'm going to be referring to throughout the talk as we go forward. And so as I mentioned, many of these epithelial cells are often kind of depicted in this vacuum of space, but actually epithelia need to arise in the middle of a very complex set of cell-cell interactions in the middle of an embryo, and even if these cells can connect correctly together to their neighbors, these have to connect correctly across an entire tissue in order to support life, and also have to connect in between multiple tissues. And I think one of the simplest illustrations of this is one of the most common body plans in metazoans, which is that of an epithelial toroid. So you can make basically an elongated donut shape where we have our skin as a prototypical epithelium on the outside and our digestive tract on the inside. And these two have to actually form together to create this epithelial toroid. And so how do cells really adhere to each other, both within and between tissues, is the question that I really wanted to focus on in my postdoc, to really understand how proper connections are made. So I wanted a system that was really robust and that allowed me to investigate some of the cell biology underlying proper cell-cell connections within and between tissues. And so I turned to the C. elegans embryo and larva as a really fantastic system, and particularly the digestive tract for studying how epithelia both within and between tissues connect. And so two aspects of the C. elegans system that I want to highlight are some of the things, not only some of the things that we think about as advantages of C. elegans, including their really fast generation time, their known and described lineage, and the small number of cells that actually make up an organ. But I also want to point out that they're still pretty complex. They have multiple different kinds of tissues that have to connect in order to create this epithelial toroid. And in addition, another facet of the C. elegans system that I was able to really leverage during my postdoc is the fact that much work is summarized in these two arrows here, that both PAR3 and the E-cadherin homolong present in C. elegans are really required for the initial steps of polarization of the early embryonic intestine. And so combining these two together, today I'm going to tell you about two different vignettes from my postdoctoral work. One, addressing the question of how do different tissues connect during embryogenesis, and a second, more recent project on how potentially disconnected cysts could reconnect within one tissue. And so to start with how different tissues, different epithelial cells connect, I want to start with a movie of C. elegans embryogenesis, in which I'm going to just highlight here the apical surface, if you just trace it around the embryo. And so we can see that this has gut, this has skin, but there's a break in this epithelial toroid that gets filled in during embryogenesis, right at this connection between the intestine and the rectum, as shown in the diagram on the right. And so this connection between the intestine and rectum is really the last place in which these apical surfaces come together to really connect between these different tissues. And this is the location of a set of two cells called the rectal valve cells. And so if we just kind of zoom into this apical surface, so the rectum has this external lumen shown in green in the inset, and the gut is outlined in the gut apical surface is shown here in magenta. And if we actually look at the cells that are present in this area, we can see that there are two cells situated right in between these really bright puncta of apical proteins shown here in these arrowheads. And so these are the rectal valve cells. And so I wanted to first ask whether these valve cells are actually really important for this apical continuity during development. And are these cells really instructing how tissues connect across each other? And so to do this, I turned to a really fantastic tool that we have in C. elegans embryogenesis, which is laser cell ablation. And so I want to point out that the gut develops on the dorsal surface of the embryo, and the valve initially arises on the ventral surface of the embryo. And they still have to find each other and connect during development. And so I was able to really leverage this spatial distance between these two progenitors by doing laser cell ablation of the valve cell well before it ever interacts with the gut, and then seeing what happens if I let these embryos develop into larvae. And so normally larvae have a really nice connected digestive tract, but when I ablate the valve cell precursor, instead, what happens is this basically ends in a cul-de-sac. The rectum never connects to the gut, and these animals eventually, they never get out of the L1 stage and arrest and starve. I can show this is very specific to the valve cell because if I ablate the cell next to it, which gives rise to one of the rectal glands, these animals are completely fine, they connect fine, and they live. So now that I know that the valve cells are very important for actually completing the digestive tract, I wanted to ask, are these cells actually polarized? And that's because when I looked at the distribution of here an endogenously tagged apical protein, you can see that there's these really bright puncta of apical proteins, but we couldn't really tell which cells exactly this apical surface was actually in. And so through much work, I generated an apical specific valve reporter. And so on the movie that's going to play on the left, you're going to see white outlines of the apical surface overlaid with the green that is just the specific apical valve. And on the right, you see time series from that movie broken out. And so what I hope you can appreciate is that from the very beginning, this valve is actually polarized towards each of the neighboring tissues, and that these puncta of polarity actually come together and are the place where the intestine and the rectum actually complete this epithelial toroid. And so we can actually color code each of these apical surfaces, showing that the valve is polarized towards both of its neighboring tissues. And so I then wanted to ask how this valve actually gets this polarity. And so I first turned back to laser cell ablations and saw that when I ablated the posterior gut, I never saw a puncta of apical proteins. So basically shown here by this lack of a dot where this white arrow is pointing, indicating that the valve cell never polarized. So I know that there's some sort of information being imparted by the posterior gut to the valve cell. To get a little bit more specific as to what exactly that information is, I turned to one of the workhorses that the Feldman lab uses, which is using tissue-specific depletion of proteins. So this enables us to get around some of the very early requirements for these proteins in these very fast developing embryos, as well as enables us to deplete both maternal and zygotic proteins. And so here what we do is we use CRISPR to endogenously tag proteins with a degron, GFP, and overexpress a tissue-specific degrader. And so using the system of depleting different proteins in the intestine, I was able to show that, in fact, one of the master regulators of intestinal polarity, PAR3, this apical PAR complex protein, is actually not required to help polarize the valve, but in fact, the ecadherin is required to polarize the valve, again, shown by the lack of the puncta here indicated by this white arrow. And so the top control is a really nice control that we have available to us, in which the function of ecadherin is present in the intestine, but still lacks GFP. And so it really nicely shows just the valve-specific component of this, of the apical surface. And so what this shows is that intestinal ecadherin, but not PAR3, is really required to polarize the valve cells. And so what this means is that in order to actually finish this epithelial toroid, you have both that the valve allows a through gut to actually form and to provide a lumen that goes all the way through the animal. And we also have that intestinal ecadherin helps really polarize the valve in return. And so this interplay between these two different tissues is really important for completing this digestive tract and leading to one continuous apical surface that can properly support digestion. So now that I've told you a story about how neighboring tissues interact to form one continuous apical surface, what actually happens sort of within one tissue? Can we actually recapitulate parts of development by looking at sort of disconnected cysts that reconnect? And so this question really arose from considering how the initial embryonic intestine forms. And so I want to remind you that there was a lot of work that showed that both PAR3 and ecadherin work together in parallel in the embryo to get all of the stuff that you need to build a proper apical surface to the right place at the right time. And part of the reason that we know that PAR3 and ecadherin are required for the formation of a correct intestine is that when we look at these intestine-specific depletions, or these gut minus, and let them grow up into larvae, when we say deplete PAR3, we have these really large cysts that form. And so these animals arrest at the O1 stage. As soon as they hatch, they can't eat. And they eventually starve. And so we wanted to investigate this a little bit further. And so I want to point out a little bit of the timing of when we are actually depleting these proteins. So the gut minus scenario that I've been showing you that gives you cystic intestines. We deplete PAR3 from basically sort of as soon as it appears in the gut. And this is actually in contrast to work from our lab and others that if you deplete PAR3 too late, these animals are completely fine. They show no real phenotypes. So what this means is that PAR3 is really required during this normal process of polarization. And so we ended up serendipitously getting a tool in hand to do really just depletion in the embryo and allow PAR3 to come. And so our initial hypothesis is that these would be cystic. And in fact, that is precisely what we saw. These animals that are embryo gut minus, so just depleting PAR3 during embryogenesis and the process of intestinal polarization, have these very cystic guts. But if we let them go a couple of days, we actually find that some animals are able to repair these cysts, not into a perfect intestine, but a pretty good one. And in fact, this digestive is continuous and is good enough to actually support the development through multiple larval stages to become a viable and fertile adult. So we wanted to investigate a little bit more of exactly how PAR3 is required for this process. And so just as I showed you before, I'm showing you that same timeline across the top. Green is going to represent endogenous PAR3 expression, and gray bars are going to represent when we are degrading PAR3 in the gut. And on the right-hand side, for each of these conditions, I'm going to be showing you the percentage of larvae over multiple days, with red meaning that these animals are cystic and blue meaning that these animals have a continuous gut. And so as I told you before, when we deplete PAR3 too late, these animals have already polarized, their guts are fine. But when we deplete just during this period of embryogenesis, this is where we find this very slow but fairly robust process of gut repair occurring. To show that this is really dependent on PAR3, we can say that PAR3, this re-expression is actually really necessary for this process of repair. And in addition, we can bring back PAR3 exogenously to show that in a completely different genetic background, to really show that it's really the expression of PAR3 at this later time point after these cysts have already formed that really drives this process of cyst repair. And so now that I've told you that PAR3 expression is necessary and sufficient for cystic repair in larvae, that a very obvious question that we had is, do cell-cell junctions actually promote or impede this repair? You might expect that cell-cell junctions in ECAD heron would promote based on the fact that in earlier embryos, these molecules work in parallel. But you can also imagine a scenario in which improper cell-cell junctions could in fact impede repair. And this is in fact what we see. So we have some data from published work that we weren't quite sure what to do with, in which the ECAD heron homolog actually has a little bit of a few larvae that survive at 72 hours compared to the PAR3 gut minus worms, which never ever get out of this L1 larval stage. And in fact, when we're doing more intensive imaging of these animals, these ECAD heron gut minus animals, which don't have any ECAD heron in their gut from the start of development, actually do undergo this process of repair. And this makes a lot of sense that ECAD heron could actually inhibit intestinal repair when we look at junctions in the PAR3 embryo gut minus animals, in which we see this sort of bulk of junctions in between cysts that could be really blocking their fusion. And so overall, what I've told you today is that we have this really nice system of looking at artificial breaks in the lumen that end up repairing, and this requires PAR3, and we think it's inhibited by ECAD heron. This is really in contrast to what I told you about kind of between tissues, in which ECAD heron is really required for imparting apical information to a neighboring tissue. And so what we think is that the combination of these two different contexts is really showing us that there has to be developmental regulation of adhesion during luminogenesis and during lumen formation in order to get the correct set of connections across an apical surface. And we think this is not something that is just in C. elegans, because the fact we can go from disconnected cis or disconnected apical surfaces to a connected lumen is found in many other systems, including the zebrafish larvae and multiple different examples of notochord development. And we're really excited to push this system forward to understand how individual cis change over time and to be able to investigate some of the physical forces that may drive this going forward. And so the last thing I'm going to leave you with is the idea that life not only requires lumens, but really requires continuous and properly connected lumens that may be differentially regulated depending on their tissue context and the time in which they need to actually form. So this project has these projects have really been enabled by multiple people. So I'd really like to thank Jessica Feldman, who has been an incredible cheerleader throughout my postdoctoral work. Several undergrads that have worked with me, including Avi Covers, all of the undergrads and master students in my collaborator, Melissa Pickett's lab at San Jose State University, as well as multiple community college students who worked with me to optimize a lot of the microfluidics imaging through a collaboration with the Stanford Microfluidics Foundry. I'd also like to thank my funding and you for all of your attention. So thank you very much.
Thank you, Lauren, for that excellent talk. So just a reminder to the audience that you can type your questions in the Q&A box. Okay, so we have a question from Chris Wright. The vertebrate mouth forms by breaking through the body covering in a precise sequence. There is specification of mouth region ectoderm and endoderm plus some neural crest. The pre-mouth tissue array is followed by BM dissolution, stomadeum formation, and bucopharyngeal membrane perforation. Would you care to comment on possible similar challenges posed in your system and in this for vertebrate toroid completion?
Yes. So this is a really fantastic question. Thank you very much. So I think there are some similarities in buccal cavity formation in vertebrates. One of the reasons that I really like the C. elegans system is that it has really sort of stripped down a lot of complexity that one might find in the vertebrate system. So for instance, in the intestinal to rectal connection, very surprisingly, and I'm sorry I didn't show this data due to time, we actually don't see a basement membrane ever form in between those two tissues that is then broken through. And so we think that this kind of connection, so basically the connection happens sort of like as the basement membrane is forming around these tissues. And so we think that the connection between at that valve intestine interface actually occurs really prior to basement membrane formation. And so we're thinking about this really as a way to investigate how you can bring together cells that may have polarized at different times and places in development. And really understand their interaction at that sort of cell-cell interface. Kind of in absence of the complexities of having to both break through a basement membrane and then find a new cell-cell partner on the other side of that. So I think there will be a lot of similarities in terms of how these cells actually potentially find each other, but not necessarily through using all of the same processes that you might find in vertebrates.
Thanks. I have a few questions too. So I guess one question I had was, are there more cadherins in vertebrates compared with in C. elegans, and does that affect how luminogenesis happens?
Yes, there are. There's been quite a large elaboration of cadherin molecules in vertebrates and C. elegans. And there's a E-cadherin homolog that combines a lot of the features of many vertebrate E-cadherins into one gene. And so we think one of the kind of surprising results that has come out of some of the work of a former graduate student is that we think that E-cadherin in kind of forming the intestine may not have much to do with sort of differential adhesion, or might also not be part of kind of really functional junction that's under tension. And instead, we think that it's really about some sort of cell-cell adhesion and really kind of coalescing the correct cell-cell adhesions together. And that is really what is required for the process of initial apical polarization in the C. elegans intestine. And so I think that in vertebrates, there's a really beautiful literature on a lot of differential cell-cell adhesion that we don't necessarily see evidence for in some of our models. But again, I think this really underscores that what we're really looking at is how different cells are connecting together through sort of reusing some of the same molecules, just in really different contexts.
Thank you. Yeah, that makes perfect sense. Thanks. Another question I had was, I was wondering, are there loss of function mutations in E-cadherin that affect luminogenesis, like either in worms or in humans, or I'm just curious.
Yes. So in C. elegans, as I showed you here, yes. So that when we remove E-cadherin specifically in the gut, yes, we do get these breaks in luminogenesis initially that then undergo this process of repair. In addition, in terms of in other systems, there are sort of implications for adhesion, but it gets very tricky, I think, because of the timing, because you really need adhesion to create a lot of these. You need adhesion to help create the actual lumens initially, and so what you're asking for is it can be kind of hard to tease apart exactly when in development and when in luminogenesis these adhesions are actually required.
Thanks. And one final question from do you think, from an anonymous attendee, do you think E-cadherin is instructive here, or are you looking at other adhesion molecules that might also be necessary?
So yes, that is a lot of work that did not make it into this talk because it's mostly negative data. So right now, E-cadherin is one of our best candidates for really understanding both in terms of intestinal repair and luminogenesis, as well as in the formation of the valve. But we have looked extensively at other adhesion molecules. So for instance, we've looked at fattin, many of the crumbs. These appear to not play a role in the early, or not play a major role in forming the through gut of the celiac intestine.
Thanks so much. Thank you, Emilio and Lauren, for your excellent talks. This seminar has been recorded and will be available on the SDB website next week. Please join us for next month's seminar on Friday, March 8th, when Jennifer Mora Mitchell from University of Colorado Anschutz Medical Campus and Wade Sugden from Boston Children's Hospital and Harvard Medical School will present. Thank you all for coming.